Good morning, everyone. All the families are here. Thank you for joining us today. My name is Nini Espinosa. I'm from Venezuela. I'm a research assistant in the Sea Turtle program. And together with Rob Drummond, we are advising these 8 wonderful children and to present the Seagrass Sea Turtle project.
All right, thank you all for being here. We are the 2016 Sea Turtle Seagrass Group. My name is Ethan Weiss.
I'm Khalil Ryan.
I'm Annika Goldman. I'm Amelia Patterson.
I'm Ellie Story.
I'm Ruby Spitz.
I'm Brooke Raffone.
And I'm Meg Manning.
And we're here to talk about the characterization of seagrass beds used by green sea turtles in South Eleuthera, the Bahamas. So first, what is seagrass? Seagrass is not, contrary to its name, is not actually a grass. Seagrass is more commonly related to the lily. Seagrass evolved 100 million years ago from land plants that returned to the ocean. Seagrass is different from algae and seaweed because besides the fact that it's a marine flowering organism, it has roots and veins. Seagrass serves as a habitat, nursing ground, and feeding ground for a multitude of marine organisms. There are over 60 species of seagrass, and unfortunately, most of which are on the decline due to sea turtle decline, coastal development, and natural or human-made factors.
Okay. Topping the list of organisms dependent upon seagrass are sea turtles. There are 6 species of sea turtles throughout the Caribbean, all 6 of these, and they are all recognized as threatened by the ICUN or International Union for Conservation of Nature. This green sea turtle, which is our key focus, is not only the most vulnerable species of sea turtle due to its entire dependence upon seagrass as a food source. It is also the most valuable reptile in the world and most commonly found in South Eleuthera, the Bahamas.
The objective of this study is to describe and characterize seagrass beds among different creeks. The seagrass is characterized by identifying the species of the seagrass and the algae in the beds, by quantifying the seagrass leaves and shoots, and estimating the coverage, density, and biomass in each bed. Using this information, we compared and contrasted different creeks, and with that, we correlated it with the abundance of sea turtles in each study site.
So these are the study sites we chose for our study. We are here, and the following creeks. We have Starved, Poison, Broad, Kemps, Rollins, Deep Creek, Plum Creek, and finally Winding Bay up at the top. And at each study site, we take a transect, or 25-meter long measuring tape, and lay it across the creek, parallel or perpendicular to the shore, to get a variety of seagrass sampling where the water is deeper and where the substrate is different. And we take a quadrat, pictured in figure 7, and move the quadrat 5 meters along the transect and lay it on the ground. And at each quadrat, we make a number of observations. We estimate the percent coverage or the density of the seagrass. In the quadrat, we identify the composition or the species of the seagrass and of the algae, and we estimate a seagrass to algae ratio. We also calculate the canopy height or the length of the seagrass leaves in the quadrat, as well as the depth of the water. And finally, we take a core sample and bring the seagrass back to the lab, and we separate it into above ground and below ground components, the shoots and the leaves. And after drying it, we use the wet weight and the dry weight to calculate the biomass. And having the above ground and below ground components separate allows us to see how much seagrass a turtle can actually consume from a sample. The turtle abundance data was collected from the Sea Turtle Research and Conservation team at CEI, from the teams in 2014, 2015, and this year's team in 2016. And they do this by using abundance surveys in the same creeks that we visited, and they'll go out on boats across the creeks and basically count how many turtles they can see in a given amount of time, and average this out to an hour.
So for each of our study sites, we described our variables by finding the mean, standard deviation, minimum, and maximum. Because we had so many variables in our study, we used the principal component analysis to narrow down the variables that were most influential to the structure and functionality of the seagrass beds. We then plugged those variables into a Kruskal-Wallis test to show how much they actually impacted our study. In our 8 study sites, we found 3 species of seagrass: turtle grass, manatee grass, and shoal grass. This is our results from our principal component analysis. The red lines, the closer they are to the axes, the more influential they are to our study. So we found that canopy height, coverage, and shoot density were the most influential. This is a very similar graph, but shows all of our observations. So each of these blue dots represents an observation from a study site.
So this graph shows canopy height, which is one of the variables that best describes the dynamic of the seagrass bed. As you can see here, on the x-axis we have study sites, on the y-axis we have canopy height. And what canopy height is that when we go to the creek, we measure the tallest piece of seagrass that we find in our quadrat, and then we take that measurement back. This graph shows coverage percent, which is when we lay down the quadrat and we look to see about how much seagrass there is in it, and from there we get a percentage, which gives us a better understanding of the density of the creek. And so again, as you can see, Winding Bay has the highest coverage percent, while Plum Creek has the lowest. This graph shows shoot density, which is number of shoots per meter squared. Again, Winding Bay had the highest shoot density, while Plum Creek had the lowest. This graph shows composition or species variation. As you can see, turtle grass is shown in green, shoal grass in blue, and manatee grass in orange. Again, Winding Bay had the highest, while Plum Creek had the lowest. This graph shows turtle abundance, which is, as Amelia described, how many turtles. You can see on the y-axis we have number of turtles per hour, and that data we collected from the turtle team. Contrary to what you've been seeing on the previous graphs, Winding Bay does not actually have the highest turtle abundance. That goes to Rollins Creek and Starved Creek.
So, seagrass communities are known as one of the most dynamic and productive ecosystem engineers worldwide, and they act as ecosystem engineers as they essentially filter sand and prevent it from covering coral reefs from sunlight. It's important to understand the composition and structure of seagrass beds in order to determine the health of the ecosystem and to improve management practices. So, in comparison to previous studies, there has been shown to be that seagrass varies seasonally, and due to environmental reasons such as temperature, nutrition, and sunlight. And in our research, we were unable to see that because we only conducted our research in the spring semester. Also, there's been shown to be a dominance of one species, and for us, that was turtle grass, as is found in all of the creeks we went to, and it was most abundant. And lastly, there's been shown to be a relationship between the substrate composition and the percent seagrass coverage, in that seagrass beds tend to thrive in sandy and silty bottoms. And this proved to be true because in Winding Bay, there was a sandy sea bottom with the highest percent seagrass coverage, and in Starved Creek, there was hardly any seagrass, and there was a rocky and rubble bottom.
So in our research, we did not find a correlation between any of the variables and turtle abundance. However, there is a known relationship between the two. This is because sea turtles depend on seagrass as their main food source, and when they do this and when they graze on the seagrass beds, it enhances the productivity of them because it prevents things like hypoxia or self-shading from happening. Also, it has been shown in other studies that biomass and shoot density are directly related with seagrass because turtle abundance, because in the places with the highest percentage of turtles, there has been the lowest percentage or lowest amount of biomass and shoot density. And it's important for us to conduct more research and to gather more data because, like Brooke said, we were limited to only the spring, and this is baseline data. So in order for us to actually find this correlation, we need to take more core samples and to go to more creeks in order to establish trends seasonally as well as annually. So a lot of information has been thrown your way, but here are some of the main things to remember. Seagrass is on the decline and is an essential part to the food web, as you can see in this picture here. Because of this, we need to focus all of our efforts on plans and management of these beds to not only protect this organism, but also the entire marine ecosystem. So here's some of acknowledgements. Big shout out to Nini and Rob as our research advisors, and thank you to all of the sea turtle research interns for helping us do a lot of our field work. Also, thank you to Earthwatch Institute. Here's our literature cited. Thank you. Are there any questions?
Can you explain what you mean by hypoxia?
Yes.
So she asked what we mean by hypoxia. So hypoxia can be caused by self-shading, which is when the seagrass overgrows and blocks out the sun from being able to reach all of the seagrass, which needs to photosynthesize. So it's like the lack of oxygen in the seagrass, and that can lead to the whole seagrass bed being destructed and dying.
Were there any discoveries or findings that were surprising to you guys?
So the question was, were there any findings that were really surprising to us? And I think part of it was just that we didn't collect that much data, so not finding a correlation was kind of surprising, at least to me. And I think especially the amount of seagrass that was found in each creek. A lot of the creeks that we went to, there's a very low percentage of seagrass, which is interesting when there was a much higher percentage of turtles that actually go there. So I guess that was most surprising.
You spoke about the seagrass as being a main component of the turtle diet, but I always heard that it was that they ate jellyfish. Do you know? I mean, this isn't your research, but I was just curious if any of you knew what percentage that was, and that these plastic bags were a function of, they would eat that and bowls of jellyfish, which was a factor in that.
So the question was kind of about sea turtle diet, and I think some sea turtles definitely do eat jellyfish, and we were focusing on the green sea turtle, and also after their juvenile stages, so when they're a juvenile, they kind of eat whatever they can find, which could include jellyfish, but once they mature, they just eat seagrass, at least for the green sea turtle, and you'll also learn more about that in the sea turtle presentation as well.
Do the mangroves have a role in the life of the juvenile sea turtle?
So the question was, do the mangroves play a role in the lives of the sea turtles? So yeah, they grow up in these bays and creeks where the mangroves are growing, and so that provides a habitat for them and a nursing ground for them as well.
I may have misunderstood, but I thought that the young man said that the green sea turtle is the most valuable reptile. And if that is what you said, please explain that.
In one of our data sources, we discovered that the sea turtle is the most valuable reptile in the world due to an unknown set of factors, but it is stated that it is the most valuable reptile in the world.
Valuable in what way?
It wasn't particularly specified in the thing that I read.
We also learned that green sea turtles are an ecosystem engineer, and like I said earlier about the self-shading, turtles are super important to prevent overgrowth of the seagrass, and if the seagrass dies, then there's a lot of other organisms that rely on the seagrass that could be seriously endangered because the seagrass.
Just one more thing to add to that. Green sea turtles also are very valuable to the culture of a lot of places. And so a lot of places eat them. Their shell is used as jewelry, or it's very beautiful. I don't know if you've seen one. And so that kind of relates to the cultural importance, as well as their relation to seagrass, and seagrass providing habitat for that, and especially in, I believe it's South America, they're extremely culturally important.